Good morning, everybody. Welcome to MolVA 2021. It's actually our fourth MolVA already, so we are looking forward and hopefully we'll have a fifth MolVA next year. So probably we'll have then a small celebration. So this is our workshop on molecular graphics and visual analysis of molecular data, taking place virtually in Switzerland this time, in Zurich, in the beautiful city of Zurich. It's a little sad, of course, that we cannot meet there in person, but probably next year the pandemic will be over and hopefully we can meet them in person again. So this is organized by my colleagues Jan Büsker, Michael Krone, and myself, Björn Sommer. And we are happy to celebrate with you today a small program. So we have a couple of papers selected. So we have 4 full papers this time and 1 short paper. The important thing this time is, in contrast to the previous MolVA, that we have a special issue on the computer and graphics journal. So special thanks go here to Joachim, who did a great job in helping us to make this happen. So this means that we indeed this year have four full papers on CNG and 1 short paper which will be published in the regular issue of the EuroVis proceedings. And more infos will be later on delivered to you by Jan, because there will be also some opportunity for authors to submit to the special issue after the conference. And then, of course, most importantly, we also have to announce our program committee. So here, I will not read it out to you, but of course, these people are very important to make everything happen. So some of them, for example, Mark even gave a talk here already at MolVA some time ago. But thanks a lot to all of you for supporting us, for doing the review process. And as you saw, we had a couple of papers. So it's, of course, this is only possible with our IPC. So let's have a quick look at the program before I will hand over to Michael, who will introduce the first keynote speaker today. So we will have 1 invited keynote. We will have then in the morning session 2 CNG papers presented, and finally we will have also a short paper presented to you. So please check the program, it's all online. Then we will have a quick coffee break from 10:40 to 11 a.m. And then we will have the second session from 11 to 12:40, which will be shared by Jan Büsker. So he will, in this session, there will be 2 CNG papers and 1 more invited talk. So this invited talk will be given by Barbara Koslikova. So we are looking very much forward to that. So let's kick off with Victor, and I will hand over to my co-chair, Michael.
Yes, thank you, Björn. So it is now my immense pleasure to introduce our first keynote speaker, Victor Guallar. Victor did his PhD in chemistry jointly at Autonomous University of Barcelona and UC Berkeley, and after that he was a postdoctoral researcher at the renowned Columbia University in New York. He then became an assistant professor at Washington University School of Medicine before, in 2006, moving back to Barcelona to become a professor at the Barcelona Supercomputing Center, where he's now heading the electronic and atomic protein modeling research group. His lab is doing research in the areas of computational biophysics and biochemistry, for example, on enzyme engineering, and he has also created the well-known protein ligand modeling software Peltier. In addition to the algorithmic development, his group is also working interdisciplinary, and that's also why I think it's great to have him here today. So they are, for example, working on data mining and machine learning algorithms, as well as on molecular visualization techniques. The title of his keynote talk today will be "A la carte biomolecular design, algorithms, and supercomputing." And with this brief and, of course, very incomplete introduction of Victor's achievements, I will hand over to him now. So thank you for being here, Victor. Thank you for agreeing to give this keynote. I think I can say that we're all very much looking forward to your talk. And in a couple of seconds after we do the handover, the stage will be yours. So thank you.
All right, so thanks again for inviting me here, and I hope I can brief you a little bit about the type of biomolecular simulations that we do, but also a little bit of a perspective of how I see the field, where the field is going, and where it comes from. I understand that while you guys are working on molecular simulations, I understand there's a lot of students here that probably don't have too feedback on proteins and molecules. So I'll try to prepare some. I prepared already some slides on that. First, where I came from. So we are Barcelona Supercomputing Center. That's the national Spanish supercomputing center. And we have around 750 employees right now. We are growing significantly. It's a new center. It's not a new center, but it's a recently new center since 2006. Basically, we, as a national supercomputing center, we provide computational resources to all of Spain and some of Europe, since we are one of the tier 0 computers in Europe. But we also have research departments like earth science, engineering, studies, computer science, and the life science department, which is the one that we are at. And indeed, these departments are quite large and are quite active, and we are the 3rd Spanish institution in EU funding, for example. And if we normalize by the number of employees, we are largely the first one, the top one. So it's a center that really prioritizes research per se. So let's start a little bit on background. Let's place proteins and ligands into the picture. I mean, you guys know more or less, but let's remind. Proteins is a chain of amino acids. Each amino acid here is like a sphere, but each amino acid is about the size, if we look at its atomic content of a molecule, like here in the right. So that will be, it's not an amino acid, but will be characteristic of an amino acid site. So imagine all these molecules, all these atoms, making these large chains that they have a lot of interactions between themselves, hydrogen bonds, etc. And they tend to fold into the secondary structures like this helix or this beta sheet. So that will be the secondary structure, things that are present in most of the proteins. So it's a repetitive structure. And these guys then fold in a tertiary structure. And this is basically the fold or the structure of the proteins. There's about a few thousands of those. So there's a lot of proteins that share similar structure, and those tertiary structures still can fold among some of them into quaternary structure. So groups of these proteins will join together in a non-bonding, non-covalent bonding, but will join together with some non-bonding forces and will make this quaternary structure. And on the other side, we have ligands or small molecules. Remind that one of these ligands will be the size of a single amino acids, and proteins are on the order of hundreds to thousands of amino acids. So very small molecules on 20 to 50 to 70 atoms. And the important part is that the proteins and the ligands do interact. So here we see also some different types of visualizations on proteins and ligands. Again, ligand very small, protein very large. For example, here we see a surface representation of the protein where we highlight in red the active site. And that's the site where the ligand binds, and through this binding is where all happens. So we will see that proteins are basically responsible for every single function in our body, from structural, from mobility, from chemical reactions, etc. And these functions happen because this protein-ligand interactions. Otherwise, the proteins are just waiting for this to happen, to these ligands to bind, to start doing something. And while we have here a surface representation, let's remind that, and the ligand in a tube atomic representation, let's remind that the protein also has atoms. Here we see another typical representation with these ribbons, these kind of ribbons that go along the skeleton of the protein. And the ligand in this colorful atomic tube or sticks representation, and some amino acids of some side chain, some atoms of the protein also shown in blue. So as I said, proteins are responsible for almost everything. They are like chemical reactors, and they do all the biochemical processes. I mean, you guys ate, and now all breakfast, hopefully, and then all this is being now processed by enzymes in your stomach. You are seeing the screen because there's a protein in your eyes that captures the light and transmit this through some proteins that transmit neurotransmitters from one neuron to another neuron to your brain, etc. So everything is doing that, and your muscles are being flexing because some protein is consuming ATP, and that's going to give, and that gives the motion of your proteins in the muscles. And because that they are the main target in pharmacology. So if you have a sickness or you have headache, you want to block these neurotransmitters that give the signal of the neurons to neuron. And then, people in the pharma industry have designed small molecules, have designed inhibitors that will block the natural ligand that creates these chemical reactions and will, for example, inhibit it. As I said, proteins are responsible for every biochemical process. So, for example, cell proliferation, cell maturation, and cell death. So if you want to kill some tumor cells, for example, you will try to accelerate this death process or diminish the growth process in your tumor cells. And that's, again, done by some small molecules that are designed to inhibit some pathway in these cells. So they're responsible for everything. And because they're chemical reactions, because they can do chemical processes, they're very interesting as well for industrial biotechnology. So we can do all the traditional chemical processes that happen in very large companies that are polluting because they're giving metals, heavy metals into nature, or because they need to use very high temperatures for this process. All these things can be done in a green manner using enzymes. But these enzymes needs to be tailored, needs to be optimized. So all these things we're going to be talking a little bit today. So if we want to tinker with proteins, we need to understand the interaction of the proteins. So we do that thanks to know the structure, thanks to crystallography or NMR. Up to now, thanks to them, I mean, we just saw the deep learning solutions of Google a couple of years ago that we are all waiting to get our hands on, where just by machine learning, they can solve the structure of proteins really accurately. So that's kind of a revolution. And again, it's not public available until we are hoping to get our hands on it. But in any case, we have crystal structures, NMR structures, so we know the relative position of these ligands that are so important because they can inhibit or they can accelerate the process and the protein. And so we know the structure, and now we can make some mathematical functions, very simple mathematical functions, that given the position of the atoms, we can get some sort of energies. And now with these energies, we can quantify this interaction. All right, and that's very important, being able to quantify this interaction. Why? Because, for example, here we see two inhibitors, two molecules that have been designed by some experimental lab or some pharmaceutical company to inhibit this protein. And we see, for example, that one inhibitor is 700 nanomolar and the other inhibitor is 7 nanomolar. What does it mean? 700 nanomolar means that you need 700 nanomolar is just 10 to 9 molar. It's a quantity of concentration, how much ligand you need to inhibit the protein efficiently. So the first one is smaller, it's not too selective, and it needs more quantity to inhibit the protein. The second one has only 7 nanomolar, so you will need much less. And because you will need much less of the molecule, you will have less secondary effects, you will have more propensity of this guy to be more specific for this target, and that's more ideal for pharmacology. And you can see how by adding this blue ring here, which is a phenyl ring, interacts very nicely in the crystal structure, and you can see that you have gained some interaction with the receptor, and therefore that's why you have a 7 nanomolar and much nice thing. So the question is, can we help finding these molecules that will bind and inhibit it in pharmacology? Or can we help to engineer enzymes the same way that you work with the molecules? You can engineer an enzyme that will do a process faster, for example, for technological applications. And also, can we help to optimize? Can we, by computer simulation, for example, told the guys, okay, add this other blue ring here, and by adding this, you will gain potency. So we can make their search much more efficient, fast, and so they will use less experimental resources. We will make it faster and cheaper. Again, I said here we look at before we just look at the molecule engineering, but we can also do the same thing with enzymes. Sorry. We can engineer enzymes. For example, here on the top picture, we have an enzyme that had a very open active site, and that's not good for stability because water molecules can diffuse a lot into the active site and then can start breaking apart the active site, for example. So people have designed some mutations of this enzyme to occlude it, to basically close the active sites, and that has improved significantly the thermostability. And now it's an enzyme that can work at higher temperatures and will most likely produce more processes, more enzymatic cycles, and will be better for industrial technology, for example. Or here we see how these people, by inserting 3 mutants in the active site, in these 3 residues M, V, and M, methionine and valine. So by introducing these mutants, they have enhanced significantly the activity of the process. And that's what industries want. Faster processes and more enzymes that work faster. So instead of producing 1 ton, they produce 2 tons or 3 tons in the same amount of time. And of course, that's bad. And that's the kind of in the field of enzyme engineering. So can we help them? Can we do this in silico processes to help them, to help engineer ligands, new drugs, to help engineer new enzymes for chemical reactions? And can we take advantage of supercomputing power? Can we take advantage of newer algorithms and better computational resources? So let me give you a little perspective of how this is working. And we had in the 80s and 90s very good pharma industries were really believing that this was going to solve all the problems, and that they were going to get these algorithms that were going to go beautifully. Then we get in the 2000s, they were very sad because the algorithms were not giving what was promised, and they were closing all the departments. I remember my PhD mates in the 90s, they were like going into work into pharma companies, sometimes even not finishing the PhD because they saw that was the future. And then all in 2000, they were fired. And then I remember this talk by Rich Frischner in 2012, more or less. It was in a conference. Rich Frischner is the founder of Schrödinger, the biggest company of modeling that tries to help these industries. And he was saying, okay, guys, you need to invest 5 more times. You need to put much more computational power. And in the first row, there was the CEO of Pfizer, CEO of Sanofi, of all these big pharma companies. And they said, no, come on, you just want to sell us more. We don't believe in your techniques, etc. He said, okay, I'm going to make a company. And he was playing bridge with Bill Gates, and then he convinced Bill Gates to invest 10 million euros, $10 million in his company. And they made a company to do that, to do the research on drugs just using computational techniques. They really solve a lot of problems. Like they make this giant company now that sells projects for thousands of millions of dollars. And around 5 years ago, let's say all companies start investing again in computational modeling. And now there is, I would say, not a single molecule that's being designed in a pharmacological company that doesn't use heavy computation at the beginning. And the perspective with machine learning, with artificial intelligence is even better. So as I said, this was a company called Nimbus that they formed, and they just sold for $1 to $2 billion therapeutic program a couple of years ago. So he showed proof, and he showed that using computational techniques and massive computational techniques, a lot more than people were thinking of using, they could address all these problems. And of course, involves as well all the software. It has been a lot of development in software in the last few years. Softwares have become much more accurate. And I'm going to show you a little bit of our 5-cent solution that we try when we try to address this problem. So this is a software called Peltier that we developed around 15 years ago, and we have kept developing in the last few years in our lab, where we basically were thinking out of the box. We were changing entirely the way that people were doing. This is a Monte Carlo software. So Monte Carlo tries to do a lot of repetitions, of random repetitions, and then say, okay, could this be the solution? Could this be the solution? And then you select or reject based on energy terms. But people were doing millions of steps, of very, very fast steps at Monte Carlo, where the motion was very slow. And we said, okay, let's change this completely. Let's do very slow Monte Carlo steps, very complex Monte Carlo steps. Instead of millions, we are going to do only thousands because they're very complex. And since we are going to be adding this complexity, we can add a lot of experimental information into the algorithm and make it more robust. So how does the algorithm work? Very, very quickly, because you guys probably are not too interested in it. But the algorithm, for example, if we look at a protein and a ligand, the algorithm introduces randomness because it has to be Monte Carlo, it has to have this Markov chain. So introduces randomness about the ligand by translation and rotation of the ligand. Also, if you can see, the protein has all these arrows in the backbone. So we use normal modes that can come, for example, from a principal component analysis of several crystal structures. So introducing experimental information in there. After we perturb the system using this randomness, then we relax the system using sidechain predictions and minimizations. And that relaxes the system, and that allows for a very effective, complex mapping of the complex energy landscape exploration. And I'm going to show you some examples of this. For example, let's look at this global search here. Also some interesting visualizations if you guys are quite into it. So here we see a nuclear hormone receptor. This is a study we did with AstraZeneca, and how an hormone, this ligand, is trying to find the active site. So the guy doesn't know where to go. He's just randomly trying to go to different positions by translation rotation, and you can see also how the backbone of the protein is moving. Now we see a close-up of the active site, and we just go back in time. And you will see how the guy will find the active site. The molecule that's inside, it's the crystal molecule, and it's there just for reference. It does not exist in the simulation, just for visualization reference. And you can see how the guy finds the active site quite efficiently, and actually will match quite nicely the crystal structure. This was not able to be performed like 6-7 years ago. And with this Peltier software, we are the first one to do this in very, very complex systems. Still requires a lot of computer power. Here I'm generous because I'm giving you an advance that we develop afterwards, but the simulation of this required like 48 hours and 512 computers. So that's you know, a computing course. That's quite a lot. A little bit simpler is the local exploration. Here we see HIV protease, for example, and the ligand, how locally interacts. And that's what they need when they design pharmacology. They need to see how the ligand, the drug, the putative drug, or the candidates for drugs that they have, will be placed inside the active site, and how it will be moving, how it will be adapting new conformations, what will be good, what will be bad, and they can do the designs of new molecules. And based on this analysis, for example, you can get how do we analyze this thing? So we, for example, plot some metrics, some distances, some RMSE, root mean square deviations, etc., along interaction energies in the y-axis, so how good it is, how bad it is, and then we apply a lot of clusterization techniques to select different binding modes, and we can provide these binding modes to the users and say, okay, this will be good, this will be bad, etc. So another thing that now is very important is to add machine learning to improve all these things by having machine learning. So for example, here we see an evolution of how far it is from the crystal along a Peltier simulation on steps of 512 processes. And you see that it takes about 300 steps for him to find the active site. And that, you know, it's about 24 hours. By adding an effective, an efficient machine learning sampling, we have really improved this thing. So basically how this works. So you stop the simulation at earlier times, here as early as only 5 steps, so very, very early in the simulation, and you clusterize. You see, okay, let's see where the ligand likes to be. So, you know, the ligand has really explored all this area, but has very few explored the area that's at around 7 amps, and that's trying to get inside the protein, but it will not get inside the protein. So now you can start more processes from this area in the next epoch. So you run some iterative epochs, and at this time, using a complex algorithm, actually it's an algorithm from the machine slots from Las Vegas, you will be redistributing the processors more efficiently, and you can see how now in only 20 steps you do find the active site, you do find the best position, which, you know, you require 300 steps, and now this can be done in a half an hour. Using again supercomputing, using again a lot of processors. But now we have really, really this fast answer. This, for example, has been done to find the resistance, and in the biomedical applications, find the resistance of the patients of HIV into medication, to drugs that are commonly given to them, Amprenavir and Darunavir. The doctors ask us, can you, if you are so good, can you predict the resistance of the patients to these drugs? So for each patient has a different sequence of the virus. We construct a model of the protein. We run the simulation with this Amprenavir and Darunavir. Then we take a snapshot of the simulation and we compute an average interaction energy. And we compare it with a sequence, let's say the wild type, a sequence that we know it's not resistant to the molecules. And by doing so, we can now plot, for example, all these numbers here at the bottom plots are each of the patients. I have to emphasize that this was a blind test. The people from this AIDS Institute passed us the patient sequence, but they didn't tell us whether the patient was resistant or not to the medication. And when we plot the results, they say, okay, these patients are resistant in red, these patients are non-resistant in green, and some patients are a little bit resistant in yellow. And we can see a very nice progression of interaction energies with that. So we can really nail down all the guys that are resistant, having a difference in interaction energy, for example, above 5 from the guy that's not resistant at all, the reference guy. And the same thing for the other drug, for Darunavir. So using these techniques, now you can do this personalized medicine. You can tell a patient, okay, don't take this drug because with your sequence of virus, it's not going to do anything for you. All right? And that's actually now a server in the vaccine computing server, and doctors from all the world can connect and can use it if they want to. So let me give you another things of the types of simulations we are doing. That's also quite visually. So for example, you want to improve a drug. We saw before a drug that by adding this phenyl ring, it was much more efficient. So we can do that with hundreds of different fragments, and we can grow them using this adaptive technique. So now in epochs, in each epoch, basically we grow a little bit more the molecule. And you can see here how we are growing this orange ring here. And by doing that, you can see how this water molecule moves, how the backbone side chains of the protein of the receptor are moving. So we can do this quite efficiently in a software that we designed last year that's called Peltier. And actually, this software is being used a lot through our spin-off into a lot of pharmaceutical companies today to do what they call the hit-to-lead optimization. So go from a molecule that can be interesting to something that gets really interesting, much more affinity, much more specificity for our target, etc. Let me just show you the video how we do it. You see, everything is a ball compressed at the beginning, and we just expanding, and by expanding it, everything is moving around and having some flexibility. And when we did that, basically we tested with against FEP, free energy perturbation, which is the gold standard in the technique. It's a very, very expensive software by the Schrödinger company. It's like €500,000 for one single computer license every year. And we got results that are almost analogous to them. We tested against the same 5 systems that they do it, and we can do basically the same answers they provide in a much more efficient and fast manner. So again, new algorithms improving, using very efficiently supercomputing by these machine learning models of adaptive, etc. So now, for example, we are applying these techniques of growing into finding what we believe it's very important. People are looking into SARS-2, but what we need to find is the medicine for SARS-3. So what we see here is a comparison of the main protease of MERS, SARS-1 and SARS-2, MERS in red, SARS-1 in green, and SARS-2 in blue. So these are very similar molecules, very similar active sites. The protease that degrades proteins, degrades the proteins of the virus, breaks the protease of the virus for the proteins of the virus to be formed for maturation. And if you can inhibit that, you kill the virus, basically. So what we are doing is all these subcavities that we are here, we are collecting all the different fragments that we can go, and through Peltier, we are growing them, and we expect to do this simultaneously for the 3 receptors. We are doing actually that simultaneously for the 3 receptors. This is something we started this year. So right now we have one molecule that binds okay into SARS-1 and SARS-2, but nothing exceptional yet. But we are using supercomputing, and we expect to have much better results in the future. But an example of how we can apply these things. Machine learning. We have a lot of it's a revolution. I mean, in 10 years, everything will be done by machine learning in the drug field. Not entirely by machine learning. I hope it's going to still combine machine learning and molecular modeling, as I'm going to show you. But it's much faster, and the results are being shown quite good accuracy and a lot of promising results. So simplest machine learning things that we are using. You have a training set, a set of inhibitors, a set of molecules that are known experimentally to be good for a receptor, to be capable of inhibiting a receptor. So we extract descriptors, fingerprints, we extract information of these guys. And now with this information, we train a model, neural network, super vector machine, if you are doing a classification, you name it, depending on what you want to achieve. And now this is super fast. This only has information of molecules, what we call 2D AI, but is extremely fast. And now you can virtually screen 1 billion molecules. So we have developed the software. Our spin-off has developed the software. I mean, not everybody can do 1 billion molecules a day, but we can, and we are trying to provide this into pharmaceutical companies. So imagine 1 billion molecules can be screened for your target a day in an approximate manner. I have to insist that, of course, this will give you an okay enrichment factor in the top 10,000 molecules, in the top 50,000 molecules, and then you might need to filter that. But there's nothing else today in the market at any level, experimentally, computationally, anything that can screen 1 billion molecules in a single 24 hours. And that's something that people were dreaming 10 years ago. So it's very important, because if you really want to find the good molecule, you need to go to these sizes of libraries. So what are we doing? We are going beyond this. We are saying, okay, we have this software that can do this very nice local and exploration of the protein and the ligand. We know this works really well with our software. We can describe what is good, which will be this red guy here. It has a very nice, in this case, is a SASA, a solvent accessible surface area. So the guy binds very nicely deep in the protein with very good interaction energy. And we know, because we compare with some crystals, that actually the right binding mode. But the same ligand has bad binding modes, a position of the ligand in the active site that's not so good, like the brown one, let's say has flipped 180 degrees, or it's a little bit outside the active site, and that's bad. And we need to give this information into the system. We cannot just provide to the system what is good, as you would do when you will be training using crystal structures and unknown experimental affinities. We need to tell him what is good and what is bad. And what is bad is very difficult to obtain, but with Peltier, it's now quite fast. So we get a set of 20 molecules of known affinity. We're exploring with Peltier, we know what's good, what's bad. We pass this into different descriptors, topological descriptors, and here it's not only 2D, here is a 3D. We have the receptor and we have the molecule. So topological descriptors, graphs from the protein-ligand interactions, images, who knows? We are still working on all these things. We make ID models, and now we can have a scoring function that will be specific for this target. So this is done on a target fashion. So one target, one set of known inhibitors, one set of simulations for these known inhibitors, and specific training for this target. And that's the numbers we're getting so far. Just 2 months ago, the correlation, the R2 was on the 0.2 values. Now we go to 0.6, which is, we are improving a lot, but we still are looking for the best clusterization. Maybe some, not always the simulations are as good as here. So maybe we need to have some identification of the bad ones by just imaging, and that will be doing it much faster in automatic manner. So those things that we have to look into. But we are pretty happy with the 0.6 correlation, average errors of 0.6, 0.7. It's not too bad. And this here I'm showing just results of the best algorithm that we had so far. And that we are trying many more things. So let me just give you 5 cents, and with this I will finish. I think I'm pretty much, I have like 5 minutes if I'm not wrong. So let me give you, or a couple of minutes actually only probably. Let me give you my 5 cents on enzyme engineering. So the same simulation we do for ligands, we can do for enzymes. Let me show you why. Here we have 2 enzymes, MMP4, very active towards a given substrate, so an enzyme that degrades one substrate. And this is MMP4 simulation with this substrate. So we can see, okay, where do you substrate want to bind? This is an enzyme that uses a heme group, which is this cofactoring here. But we do the simulation of this kind. All right. And then we find this binding site. Okay, that's nice. We have this binding site. That's what we believe. It's our best assumption of how this substrate binds. And in our simulation, the enzyme should degrade this substrate by this position. And then we have MMP6. It's an enzyme that does not have any activity towards the substrate, 0 activity. And if we look at this MMP6 guy, it's this other guy in blue, all right? It has 2 different residues in this position. It doesn't have these 2 residues, histidines, that will be creating this electrostatic anchor and will be creating this nice interaction. That's the residue. So asparagine and glycine, 2 different residues. Okay, so now we have information of a guy that's good and that react, and information of a guy that does not react. And actually, this was a command from an experimental lab friend of ours that they wanted to mutate this guy because this guy is very stable, as opposed of MMP4, which is not stable, and they wanted MMP6 to be reactive and be stable. And they always thought that the chemistry should happen where the mutation was, with this kind of C-terminal, this pink tail that MMP6 adds, that MMP4 does not have. And we told them, oh, sorry guys, nothing happens there. It happens a little bit far away from here. And we recommend you to take these 2 residues, which are exposed in solution, which are residues that you will never touch, and change them to histidines. Of course, they didn't believe us. They were reluctant to do that, but at the end, we convinced them to do that. And by doing these 2 mutations in the surface, now we have an enzyme MMP6 that's very stable and that reacts almost as fast as MMP4. So that's the things that simulation can bring you, for example, in enzyme engineering. Very nice type of modulations. We can train mutants, and now we can do the simulation MMP6 with the mutant, and we observe basically the same results. And tinkering enzymes for better activity. We do the same thing for thermal stability, for several things. I would probably skip this second example, which basically, if you remember, I told you that an enzyme has an active site and has one chemistry, and that's the rule. Enzymes have only one active site. Everything else is structurally designed to hold this active site, to make interactions, etc. But now we have been able, with this diffusion of these ligands, to select additional binding sites, other areas where the ligand likes to be, and we can turn these binding sites into active sites. So now we have enzymes with 2 different active sites, so that we can do twice as much as reactions. And actually, we can even tinker one active site to be doing a different chemistry. So now we can have one enzyme that does 2 different chemistries, and we can make complex cascade reactions using only one enzyme. That's a huge advantage. And actually, the number one company in enzyme productions with like 55% worldwide shares is asking us to do some of this for them. So that can be small revolution in industrial enzyme engineering. So let me just finish, but I mean, you've seen a lot of different visualizations. You see that it's key for us to understand these things. It's key for us to extract data. It's key for us to manipulate this data and to have a very quick as possible understanding of what's going on. And we have been working actually a lot with Prevost from UPC in the Polytechnic University of Catalonia in better ways of visualization. I'm sure you are more aware of this because he probably has introduced it or you've seen this kind of papers. And you know, things that we can recognize which amino acids are a nice interaction with the ligand, where we can highlight the ligand, etc. But actually, I want to show you a little bit. Let me see if I can do this. Where we all want to go. We all want to go to this. All right. We all want to go into this virtual reality thing. We want to be able to manipulate. These are, I mean, this is actually a company from the United States, Nano. And this is actually we talked with Prevost like 5-6 years ago that we wanted to do. Well, bad news, someone else has done it. Doesn't matter. There's many people working on these things, but that's what we would like for a better understanding. A cooperative working of 5 groups in the world working with the same thing. So one specialist in Singapore knowing a lot about this molecule, helping you to design these things. And actually, this is only now at the visualization level. What we would like is to have on-the-flight simulation with Peltier on top of this. So not only you will be visualizing, but you will be doing the simulations, you will get immediate answers, and you will be coupling this thing into this. So that's where I would like to go. The EU funding system doesn't seem to agree with me 5 years ago or 4 years ago. Hopefully they will change soon. So let me just finish by thanking everybody that has done this work possible. The group, basically, Barcelona Supercomputing Center. And I'm open for questions from you guys.
Hey, thank you, Victor, for this very interesting talk. Looking at the Discord channel, it's still pretty early, so we are still waiting for questions, but I have a couple of questions of my own. So as I will start with this one, and hopefully people will then join in. So the last example, and also the very nice visualizations with 3-dimensional molecular structures that you showed. I mean, this is, of course, like you said, very close to the heart of what we're doing here at this workshop and in the visualization community at all. My question here would be, do you think this all these 3D visualizations, especially the virtual reality visualizations that you mentioned at the end, is this more for communicating results, or do you see a bigger potential or also potential for data analysis on the go, so to say, during the simulation?
I think this is a very important question. I've been discussing this with clients of the spin-off, with colleagues of mine and at the academic center, and you know, how much do we invest in this to go there? Is it really going to be used? Is it enough with the 2D we have now? Or I think my personal view is that it will happen, and it will happen. You know, there's a barrier to do that. Our brain might get tired after a while but we will adapt. And if it can really provide new ideas, if it can really help us, then it will happen. I talk to people who use it on an almost daily basis, and they say they got used, and now they love it, and they really like it. Still, the problem is that, as I said, is right now is that just as the visual. You cannot interact with it. You cannot do simulations at the same time. But the day that we can do simulations and we can have all these different screens around, looking at you know, results, and then you can get these results at one point in this plot, and then from this plot create another input file, and launch the simulation, and in 2 minutes you get the results. In that day, I mean, pharmacological industry will make another revolution, and they will use it. I'm pretty sure. So that's why I wanted to bring this up to you guys. If any of you wants to do this, let's do it. We have the software that does this. We need, you know, let's ask for an ERC grant. Let's ask for a synergy. I mean, this is the future, and Europe is behind. It's clearly behind. I mean, it's sad, but that's the truth. I mean, Americans are already commercializing these things all over. This company of America contacted us 8 months ago. Okay, if you want this, we want to use this. Here you have a demo. Here we have the glasses that we sent for you. We need to do it. We need to do it, and the EU should be funding this. So if any one of you have access to the people granting panels, we need to have this. We need to have a big project with 10 million euros doing this, and we need to do it.
My second question, so we're still waiting for questions from the audience. I would just continue with my second question. So you showed a lot of exciting application from machine learning for data analysis. And of course, you also showed visualizations of the results of this machine learning algorithms like scatter plots and this. And in the visualization community, there's currently a lot of discussion going on that we should not just visualize the results of machine learning algorithms, but also create visualizations to create a better understanding of how the machine learning algorithm works internally, so to say, so how it comes to a certain result. Do you also think that would be interesting in your field in biochemistry, or do you think, or are you just to say happy with the results? And do you think people can work with just seeing the results and that not knowing what's going on underneath, so to say?
I imagine here there will be like two different sets of people. One that are going to be just using this out of the box, like a product, and they probably don't want to, or they have the capacity to play around with the algorithms and the variables. But now there is clearly in AI, there is this now we have applied for some grants, including it. There is what they call this a smart AI, an AI that you really see what's going on, and you can really understand why the answer is this one, and then you can really parameterize and tinker the AI machine. What a couple of years ago it was more like a black box, and in doing so, I imagine visualization is a key aspect. So in our group, certainly in our group, that we develop a lot of methods and we also apply them. We would love to have some visualization tool that will be connected to this smart AI thing, or I don't call it a smart AI. I don't remember exactly the name of it, but where they, of course it's a little bit slower, but they are creating human readable protocols, and actually it has a lot to do with visualization, human readable algorithms that you can see, you can really play around with it more and understand why this is happening. So and in that sense, visualization is key, sure. Yeah. So I would say yes for many groups.
Maybe a very quick follow-up question to my first question. You said that all this virtual reality interaction is, if I understood correctly, quite lacking. So do you think the visualization methods are already there, or do you also see challenges for the visualization? So would we, if we have a virtual reality system for structural biology and the interaction part, so to say, would be fixed, would be working nicely, do you think there would be challenges also for the visualization, or do you think we could just continue using the molecular models that we are using right now?
Well, molecular models are not bad, but they're not bad until someone can create something better, no? And then we oh, well, we were working with that thing. I cannot believe we were using that. I would imagine that there's a lot of challenges to be addressed. Yes, I mean, that will be my intuition. Honestly, I play around with a couple of them, and they are not that good. I mean, you see this movie, it's not like that. And for example, what we ask them always is, okay, we want to have a lot of panels because we want to have, the 2D plot here, the scatter plot here, the several things. And they say, okay, well, we are working on that. We can give you a little bit of that. Of course, it's something that But if I look 4 years ago, there was nothing, and now there is this. You know, it's in 5 years, I'm sure it's going to be there, and people will use it. Probably also the fatigue problem, the immersion system might be slightly different. The gadgets to operate it, I think they're improving most of the time. Yeah, I think both at hardware and software. I mean, it's super nascent field, and I would imagine it has a lot of challenges for you guys. I mean, probably exciting challenges. Yeah.
Yes. So it's worthwhile continuing working on this. Maybe someone comes up with something better that.
And come to me. Once it comes out, come to me. We'll implementing it.
And maybe augmented reality will also be the next thing after virtual reality.
Yeah. In this small world, I mean, we are looking at something that you cannot look at it in reality, because you cannot, you cannot see molecules. No, there's nothing to. But certainly, yeah, having some augmented reality in the conference room or holograms, these kind of things. Yeah. Anything that will give us a better understanding, an understanding that we can share with people who are remotely connected, and that will also very quickly suggest us options, because then, of course, in all this, then there is the brain, the artificial intelligence that will be learning for each session. Okay, the human has thought to do that. This group of medicinal chemists that I've been working for 2 years, they were telling me to do this, I'm learning. And then you just push a button and it's working for you and we can all retire. All these components are there, yeah.
Okay. So I guess it is still early morning. I don't see any other questions, but I also think in the interest of time, we have to move on. I would have still tons of questions and things we could discuss, but I think also you have to leave, right? So
yes, sorry about that.
No worries. As said, we also have to move on the program. So again, thank you for joining us. Maybe if you have time, you could check the Discord channel afterwards. Maybe some late questions will pop up there.
Totally. Or you can email me questions anytime, no problem.
That would be awesome. Okay. Well, thank you very much. And with this, I think we can hand over back to Björn and
have a great meeting. Bye.
Thank you, Victor. Thanks a lot for joining. It was very interesting. So welcome everybody to the second part of our morning session. And now we will have a couple of talks. So we will have 3 papers which will be introduced now. I hope you had a great time with the introduction talk, which we just heard. So we will continue now with Mathieu Noizet. So he's from the University of Durham, Champagne-Ardenne, and he will be talking about a collaborative molecular graphics tool for knowledge dissemination with augmented reality and 3D printing. So it's a very exciting work. So we're looking forward to this. So if you have questions, because the presentation is going 20 minutes, so there will be not really time to have a Q&A afterwards. Please put your question into the Discord channel, or you can also use the YouTube channel and ask questions. And Mathieu, he's also here, will be then live answering you directly. So thanks a lot, Mathieu. Let's have a look at your recorded stream.
Hello, I'm Mathieu Noizet. I'm a student in my second year of master degrees at the University of France. I'm going to present my work entitled A Collaborative Molecular Graphics Tool for Knowledge Dissemination with Augmented Reality and 3D Printing, supervised by Stéphanie Prévost and Jessica Jonquet-Prevoteau. After introducing the background and previous work, I will present our proposed tool based on 3D printing augmentation. I will describe its pipeline and focus on 3 processes: the pre-processing, the live detection, and the augmentation. Then, I will present quantitative results explaining our optimization parameters before commenting on demonstration video. Finally, I will conclude and give you some perspectives. For a long time, modelers used different representations to visualize and analyze molecules and their trajectories. This is a very important aspect in the study of the structures, functions, and dynamics of the biomolecules, but also in their exchanges with the experimenters. Moreover, the advent of immersive visualization tools has allowed modelers to better take into account the three-dimensionality and to more easily share their results with their non-expert modeling colleagues. Furthermore, the emergence of low-cost virtual reality headsets allows for 3D visualization and total immersion in the molecule to study them from all angles and under different representation at the same time. The user can use the joystick to manipulate the molecule. Even if it's a real advantage for studies, the use of peripherals for interaction is difficult to handle for modelers and even more for their non-initiated collaborators. In addition, this type of headset removes the collaborative aspect and the user is isolated. To avoid this problem, they turn to augmented reality headsets like the HoloLens. They allow collaboration and interaction is not done through peripherals, but directly with the hands, which seems easier. Unfortunately, the manipulation, usually called air touch, is again difficult for a non-initiated as they are too abrupt and not very precise in their gestures. Moreover, with the development of 3D printing and low-cost monochrome printers, modelers are again using physical 3D printing models. In this way, modelers and their collaborators find a natural and intuitive handling to study and manually explain to biologists the obtained results. This process has several drawbacks. Multicolor 3D printing is expensive. It's necessary to multiply the print to allow different representation of the same molecule, and not all representations are printable. Indeed, to help understand a molecular or docking, multiple representations are used. Sometimes there is no contact between them, which makes 3D printing of the composition impossible. Finally, the display of information around the object in transparency is not possible. We thus propose to associate augmented reality and 3D printing in order to use the printing molecule as a tangible interface and to take advantage of augmented reality. Therefore, we can display all available and desired representation of the concerned molecule. This idea has been studied before. For example, Olson's team in 2005 created a prototype to augment a 3D printing molecule as illustrated here. This paper has highlighted the potential of the information to be augmented, especially around the object. But this prototype has several drawbacks. First of all, Olson used 2D markers to detect the molecule. These markers can be placed on areas of interest and limit the possible movements because they must be always visible by the camera. Of course, we could put more markers, but it would hide more areas of the molecules. Moreover, here the visualization is decoupled from the handling. The augmentation is visible in a remote on a monoscopic screen and not directly on the molecule. This makes the handling indirect and not very intuitive, especially for a non-initiated. Of course, since 2005, Olson continues his work, but also with markers and 2D visualization using smartphones or tablets to augment the information on the molecule. In 2019, the combination of 3D printing with augmented reality has also been proposed by Martinez and al. In the context of teaching to help students understand the construction and conformation of molecules. The manipulation is natural thanks to the physical elements and the assembly of them is interactive. They use various color patterns and the detection method is by color. This technique presents for us several disadvantages. Indeed, it's restricted to the atom or group of atoms, and its detection by the color can degrade the quality of the possibilities of augmentation. Therefore, we wish to develop the concept of augmented 3D printing by turning our 3D printing object into an intangible interface. In the long term, this molecular visualization tool will allow a natural and intuitive interaction with different representations of the molecular modeling to perform a complete visual analysis. This user-friendly tool will facilitate the knowledge dissemination and collaboration between interdisciplinary researchers. Indeed, they all have different knowledge and it is not always easy to exchange, but the visualizations help them a lot, as it often is more meaningful than words. Such a tool will allow them to benefit from the advantages seen above while avoiding the disadvantages. When designing this tool, we wanted to keep a good immersion and good augmentation. The FOV of the first HoloLens, the headset chosen here, is very limited, so we chose the size of the printed molecule so that the user can see it fully augmented when he holds it. The maximum size of the enclosing volume is therefore 10 by 10 by 10 centimeters. Moreover, it's printed in white matte to prevent the appearance of the artifacts of the augmentation. We present here a first version of the tool which already allows to display separately or together different representations chosen by the user, to make annotation and to rotate around the object. In the long term, we will add the possibility to manipulate the 3D printed object and to realize a collaboration with several HoloLens. In this short-term tool, we focus mainly on the detection component and on the validation of the principle of augmented 3D printing from the molecular modeling. At the beginning, we add different representations from the works and the study molecule. From this, we choose our main representation, here a surface representation, which is used to print in 3D the object and to serve a display for the augmentation. Our pipeline is composed of two steps, the preprocessing and the live steps. In the next slides, we explain these steps. The preprocessing step consists on compute relevant features to detect the position and orientation of the object visualized in live. For it, we create a database of the in-synthesis views, called reference views, with their features. In order to maximize the quality of detection and limit the number of views, we choose to place all views around the object equidistantly as described in the demo paper. Each view undergoes the same process as the live capture that we see in the next slide. Next, we crop by the orienting bounding box the view and compute all data needed to replace the 3D model in the same position and orientation that this view. This data will be used in the augmentation process. The reference view with all its features are saved in the database. The live detection process consists in finding the closest reference position and orientation corresponding to the live capture. To separate the object from the background, the live capture is cleaned with an auto-adaptive thresholding and binarization. Then, we crop the result by the orienting bounding box. In the next step, we search in the database for the closest reference image to the cropped image. The comparison is based on the XOR operator, which takes into account the shape and is easy and fast to compute. We select the reference image where the number of pixels of its respective XOR is below the epsilon error rate. Furthermore, in order to limit the number of comparisons, we only look at images with a difference of size ratio below the beta threshold. Then, the closest reference image with these features are used in the augmentation process. The augmentation process consists in computing the rendering of the additional information and embedding it in the real scene at the right location. By mapping, we can determine the corresponding pixel of the color image in the depth map of the HoloLens. From the previously found data, we project the reference point P corresponding to the origin of modeling to the depth map to obtain its distance. To accurately determine the orientation and position of the virtual object, we compute the transformation matrix from the object coordinate system to that of the world, and then from the world to the HoloLens coordinate system. For this, we use the stars features with the reference view and the distance found by the mapping. Finally, we apply the transformation matrix to the selected representation and run the rendering. The steps of detection and augmentation white location computation, which need to initialize the augmentation, are too time-consuming to respect the real time on the first HoloLens. We have performed different tests to speed up the detection without affecting the quality of the result. To limit the number of comparisons, we begin with the size of the database. We test for n the values 1, 2, 4, and 8,000, and we compare the average of error rate for 10 captured sample images from the HoloLens by varying the printed object orientation and the luminosity. As the difference of this rate between the 4,000 and 8,000 databases is very small for a double number of images, we set n at 4,000 for the continuation of this test. Again, to limit the number of comparisons, we study for the threshold of size ratio of the cropped image, the average error rate, and the average runtime. For beta, we test the value 0.05 and 0.1 and 0.2. At 0.05, we note a degradation in the error rates, so we set beta at 0.1. To accelerate the runtime while preserving the quality, we examine the possibility to reduce by a scale factor the size of reference image in the database. When the original resolution is divided by 4, the object shape in the scaled image gets coarse, increasing the error rates. In contrast, for the 1 and 2 factor, the two found reference images are identical. After taking into account the runtime, the scaling factor is set to 2. This is a demonstration video where the molecule is scale modeling. The database includes 4,000 images and the ratio threshold is 0.1. In a real experience, the reader sees the augmented scene in stereo. However, the video capture of HoloLens is monocular, so a noticeable shift is visible. After the initialization step, the interface and the post-selective visualization are displayed. With the slider, you can change the transparency of the selected display. The user can move the interface to clear this workspace, select from thumbnails the representation to study like hair secondary structure, surface, or calcium, show one or more representations at the same time, and add annotation. Once all set, the user can move around about your molecules to search many points of view, find regions of interest to get a better understanding of the molecule structures. These possibilities are very interesting for the dissemination. The user can move the printing object if the initialization step of the augmentation is restarted. To conclude, thanks to this first tool, the molecule is detected and augmented without the addition of any kind of marker. This has allowed us to validate the principle of augmented 3D printing, and the first use feedback from biologists and biochemists show a real and encouraging interest. Now, we need to optimize the first tool and turn the object into a tangible interface. For this, we need a robust detection and tracking methods against occultation generated by the user end. We would also like to add different modes of collaboration on-site with multiple HoloLens and remotely. Thanks for your attention.
Great. Thanks a lot, Mathieu. Really nice talk yeah, really interesting approach. We don't have much time because we had exactly 20 minutes for the presentation. So, but maybe just one question and the other questions, please post it just in the chat, and Mathieu will answer you within the Discord channel, or also you can use the YouTube channel to ask your questions. So Mathieu, what are the next steps with your great approach? So are you working now more on the synchronization, or what are the next steps?
In this project, the next step is use the second HoloLens to have more interaction and intuitive interaction, because with the first HoloLens, we must focus the button, reach a little point and click in AirTouch. And with the second HoloLens, we can just touch virtually the button to interact with it. And that is very interesting for the interaction with the applications. And for the continuation in this project, we must to make a lot of interaction, and we want to move the molecules with the hands to use the physical molecules like a tangible interface. For this, in this application, for the moment, we just use the molecule, and we have just initialization step. We don't have interaction with it. And we want to set the occlusion with the end of the molecules to coincide, to move it. How I can say that? We want to use occlusion, handle occlusion to manipulate correctly the molecules with the tangible interface.
Great. Thanks a lot, Mathieu. I have more questions, and I think also the audience will have it. So please just put it in the chat. So thanks, Mathieu. We have to hurry up because we are a little bit over time already. So thanks, Mathieu. So let's continue with the next talk. And this will be given by Thomas Alderich. So he's also here in the channel. So his talk will be is recorded and will be played to you. So please welcome with me, Thomas. So his talk will be on computational design, fabrication, and evaluation of rubber protein models. So there's some relation to the previous talk to Mathieu. So we are looking forward to that.
Hello everyone, this is Thomas Alderich, and today I'm going to present you this work by the title Computational Design, Fabrication and Evaluation of Rubber Protein Models that I've been collaborating on with Daniela Giorgi, Luigi Malomo, Paolo Cignoni and Monica Zoppe. So let's start with a very brief introduction about 3D molecular visualization and tangible models visualization, and by telling you why we feel that tangible models are important for visualizing molecules. So molecular visualization software like the famous PyMol is a very important tool both for teaching and research purposes in biology and biochemistry, as it allows one to easily manipulate, explore, and annotate 3D molecular data. However, on-screen visualization often lacks the ability to convey a good sense of the scale between molecules and their interactions, especially to a non-expert audience. In the contrary, tangible models that can be held in hands and directly manipulated, like these ones that I have here, they facilitate the creation of a mental image, and therefore they can more intuitively, convey a sense of the scale and the interaction that can happen between different monomers or different molecular assemblies. And there is a long tradition in the development of such tangible models. In particular, it all started with the works from Pauling and Kanjiu in the late 60s about creation of models for the myoglobin or the DNA. And this tradition has carried on until nowadays, where it gained even more importance thanks to the availability of 3D printing and novel fabrication techniques. And this has been demonstrated by the success, for example, of the NIH 3D print database that collects thousands of ready-to-print molecular models uploaded by an open community. However, there is yet not a general solution for fabricating complex biological entities and visualizing them in a tangible way. And in this context, our goal is to provide a pipeline for the fabrication of complex molecular assemblies that are made up of soft monomers that can be easily manipulated and assembled, like the ones that you see here in picture. And in particular, we want to do so focusing on the representation of proteins at a constant scale to emphasize their scale and interaction relations, and on their flexibility, and this is why we chose to use soft materials like silicon. And we also want to use a production technique, a fabrication technique that allows us to scale in numbers, because say that you want to use these models in a classroom environment, you will need probably to distribute those to 20 students. And so you want to have an efficient way of fabricating them. All in all, what we want to, achieve is to provide a tangible metaphor to visualize association mechanisms that happen inside a molecular assembly. And so let's have a look at how of the pipeline that we propose for doing so. So it all starts with a PDB file that is a file reporting all the atomic coordinates for the atoms that comprise the molecule. Then from this PDB file, we want to extract for each of the subunits involved in the assembly a solvent excluded surface, or that is a Connolly surface. Then, since we are interested in really matching the interactions between the different subunits and this concept of, binding or interaction between two subunits in an assembly, we want to make sure that, that all the surfaces they correctly match together, and so we apply this matching subunit step. Finally, once the final surfaces are ready, we automatically design a mold for the generation of every subunit involved, and with this mold design we can then go to the actual fabrication pipeline, that is 3D print the mold, cast the molecules, and then with your monomers, with your subunits, assembly the final molecular assembly. Okay, so let's have a look at each one of these processing stages. So the first, as I've told you, is to extract a Connolly surface from your PDB file. And what we do is, using PyMol for each chain, so for each subunit, we extract an independent Connolly surface, and output would look something like this. But unfortunately, if you zoom in, what you see is that frequently, when extracting a Connolly surface independently for each subunit, what happens is that you can have small intersection between different subunits, where along the interaction sites, where the atoms get really close one to each other from the different subunits. And also, in any way, the high frequency detail that characterize these surfaces, it makes the match between different subunits harder. And so, since we are really interested in providing this good metaphor to visualize and teach the subunit assembly, we want those surfaces to perfectly match together, sacrificing a bit of, accuracy in the surface reproduction. And so to ensure this correct match, we deform the subunit shapes, the Connolly surface of the subunits, locally to the interacting sites, so that they perfectly match together and they guarantee a smooth interaction surface. And we do so going from what we call the Connolly surface here on the left to an interpolated surface here on the right, applying for each subunit an interpolation scheme that is defined on an implicit representation of the surfaces as distance fields. And this scheme works like this. So let's call dA the distance field of the subunit surface that we are processing right now, and the other the distance field of all the other subunits in the assembly. So what we are doing we are looking for a D signed A whose zero isosurface, it smoothly converges to a medial surface between interacting subunits in areas where the two surfaces are closer than a threshold T, that is in areas near to interaction sites. And in particular, in our paper, we set this T to 1.4 angstroms, that is the radius of a water probe. And then this zero isosurface should rapidly converge to the original surface everywhere else. So as you can imagine, that this t parameter, in a way, it controls how the interpolated surface will look more to the Connolly surface if you set t parameter to lower values, or setting t parameter to higher values, you will end up having smoother interaction sites and also wider interaction sites, making the assembly even easier and more intuitive, okay, but disrupting more the original surface. And okay, so the output of iterating this interpolation scheme over all the subunits is a set of perfectly matching subunits whose surface is deformed only locally to the interaction sites, as you can see here from the animation. You see, it only changes along interaction sites and all the rest remain unchanged. Okay, so once we obtain the interpolated surface for each of the subunits, we can then proceed to automatically generate the molds for fabricating them. So for whom is not familiar with molding, a mold is an object made of a set of parts, and these parts can be assembled together to form an empty cavity or hollow shape that has the shape of the object that you want to produce. And this empty cavity can be filled with some casting material that in our case is silicon. And then once the material is cured, it will take the exact shape of the cavity and so of what you want to produce. Then what happens is that you open the mold along the extraction direction for every part and free the object. Unfortunately, designing such mold is a very complex task, as it requires you to choose an extraction direction for each one of the molds, and also the geometry of the parting surface that allow you to open the mold in such a way that the object can be that the mold can be opened and the object can be released. And this requires to account for some constraints, for example, like the undercut constraint, that an undercut is like these areas that are highlighted in red here, that if you consider the extraction direction of the mold parts, what happens is that in the undercut volume, in the undercut surface, you will have that the mold gets stuck into the object, and so it will be hard to release it without damaging the object or the mold itself. Fortunately, recently we have had some recent contributions in the computational fabrication domain about the generation of soft molds for casting rigid objects, and we are going to apply this knowledge in our case, but rephrasing it and creating a rigid mold for casting soft objects. So let's have a look, a very high level look at the pipeline here that we use in this paper. And what we do is, okay, given a subunit, we analyze a set of extraction directions around it and find the pair of extraction directions to minimize the number of undercuts that are present on the surface. Since the proteins are made up of soft materials, so we allow some undercut to be present. Then, given this pair of direction, the algorithm automatically defines the geometry of the parting surface, and this parting surface will be then the base for the automatic generation of the mold design. Okay, this mold design can then be 3D printed, and the mold can be used to generate copies of the subunit. And I will show you in a moment, looking at the results, how this ends up. So this concludes the part about the computational part about the pipeline. Now I want to very quickly show you some of the results and show you the results of fabrication and assembly. And we propose for the results the molds and assemblies for 3 biologically relevant macromolecules that, among the different the millions of different proteins available, for example, over the PDB repository, we have selected to be of interest for a larger audience. And these are the F-actin, the hemoglobin, and the histones. So, for each one of these results, I will also show you on screen this QR code here on the right that links to NIH 3D print database page that hosts the modes files for the result that you can download, 3D print, with which you can try the technique yourself. So let's start by F-actin, that is a part of the cellular skeleton. And what is nice about F-actin is that it forms indefinite polymers out of a single monomer that is repeated in this double helix structure. And this allowed us to use only a single mode for generating the same subunit all over and chain it in an indefinite polymer. This is the assembly I'm going to show you. As you see, thanks to the exactly matching surfaces between at interaction sites, the molecules really snap together when you assemble them, and so it's easy to find the interaction site and to create the final assembly. Even if probably using some magnets, that is something that you are looking onto for future work, would really ease up even more the assembly and also make it more solid once created. Okay. This is the hemoglobin instead. That is a textbook case of a tetrameric protein composed of two different subunits, alpha subunits in blue and beta subunits in red. For this we need two modes because we have two different subunits. And this is the assembly. Again, you see how the exactly matching surfaces really help into snapping together the two monomers. And one thing that I want to note here is that this choice about using a single mode for replicating the same subunit that is repeated inside the assembly is a user interaction choice. The algorithm by itself would be able to produce a single mode for each single subunit involved without any problem. So in any case, even without the user interaction that tells you, okay, you can save a mode here, the algorithm would work well all the same. And finally, this is the histones models. One thing that you probably note here is that the modes they are slightly different. And this is because in case of very complex molecular shapes like this one, our rigid mold generation algorithm can fail. And in the paper we detail how to detect such possible failure by looking at the topology of the parting surfaces and at the amounts of undercuts that are introduced by our mold decomposition. And in case of failure, the pipeline outputs a composite soft mold as a fallback strategy, like this one in picture, that is based on our previous work about soft mold generation that we based our mold generation approach on. And finally, this is the assembly procedure for the histone octamer. Again, here maybe you see more why also embedding some magnets could really help, making the assembly more stable and also easier. And also you really very well appreciate the flexibility of the molecules and their dynamicity. Okay, so let's end the discussion with discussing the user evaluation in classroom that we have conducted. Unfortunately, it is a small and partial evaluation due to the COVID restriction that were involved in force in Italy during the past year, and so we weren't able to conduct a proper user evaluation. And we have done this evaluation involving two teachers from different schools. Teacher one was from a science high school with a class of students about age 17, And teacher 2 comes from a technical high school with students about age 15 that have never had any previous experience with proteins or molecular visualization or molecules in general. And we provided the teachers, both the teachers, with a full kit of our produced fabricated protein assemblies, and they were able to use them in class to, as a support, as an aid for a lecture about lectures about biology and about proteins. And after some time, we discussed and we gave some questions to the teachers to try and evaluate what was the impact on the classroom and on the lecture of using the models as a teaching aid. And we are a bit short with time, so I don't want to discuss it too much in depth, but in general we have had quite partial but positive response from the teachers, and this has encouraged us to start planning a new user evaluation and maybe a more broad user evaluation as schools will properly open again in Italy, so that we will be able to do a broader user evaluation. Okay, so to conclude, we presented a pipeline for the fabrication of SOT molecular assembly, and we demonstrated it with a set of fabricated examples, like the ones that you see here in picture. And finally, we also provide a limited assessment in the form of an in-classroom user evaluation due to the COVID situation. For our future steps, we plan on expanding our user study to have a broader and deeper evaluation. And also we plan on defining a way for the automatic embedding of magnets in the interaction sites during the silicon casting procedure, so that the magnet after the casting will get trapped inside the silicon shape of the subunit, and docking will be easier and interaction will be easier. So this concludes our discussion. Thank you for your attention, and feel free to pose any question or send us an email for any question or curiosity. Thank you for your attention. Bye.
Hello everyone, welcome to this presentation. My name is Victor, and I would like to talk about a tool that we created. It's called VRdeo, and it's a tool for recording and viewing 3D educational material with an option to export a 2D video. We realized that creating educational videos, it's a difficult task. It's really hard. It's hard to learn to use existing tools. It can be quite time consuming, even for an experienced animator, and it demands a lot of experience with animation and even some technology, or at best with the topic itself. There are some existing desktop tools that you can use to create some educational content, such as some general modeling tools like Blender or Maya, but the problem is that they have a really steep learning curve. And there are also some specialized tools such as PyMol or VMD, but the tools are meant only for exploring the molecular data, and the user can export some videos there, but it's mostly by using a command line, and it's really complicated. There are also some VR-based tools such as Mindshare or Tvori. Tvori has a lot of low-level tools for creating animations, such as setting the light, air, and other things, but it's too complex for the purposes of preparing educational materials. There are also other VR tools. There are tools for the data exploration, where you can view the data, but you you cannot create the material in other way than just record screen capture of what the user sees. So in cooperation with a protein engineering expert, we have formed several requirements for the tool itself. It should support asynchronous communication, so the communication of the content should be asynchronous, which does not require the simultaneous presence of the tutor and students, which means that the tutor can create the content that the users can then watch later. It should be intuitive and easy to control, so the user doesn't have to spend too much time learning the application, and also too much time by just simply creating the content. The tutor's avatar, including its position and gestures, should be captured in the scene, so the students can easily connect the voice recording with the tutor's actions in the scene. There should also be an option to quickly check already recorded material directly in VR environment, so you can see a preview of what you already created there. We should also give the user, the narrator, some option or some visual feedback, so he should stay visually informed when recording or replaying the content. The next requirement is that the students replaying the recorded story should have an option to pause it and then explore the data and the scene in detail by manipulating with it, and then again replay. And the last requirement is that the tool should also be able to export traditional 2D videos, which can later be shared with more students without the VR equipment. So based on these requirements, we created or came up with 3 user roles. And the first one is the narrator, who is the content creator, the originator of the story, who is recording the story. And then there is the observer, who is the consumer of the story. And the last one is the camera operator, who is responsible for recording a 2D video from the narrator's content. Based on those roles, we also created we designed the operation modes for the application. So for each role, we have different mode of operation. So for each user role, user can utilize these modes to create or to interact with the application. The first user role is the narrator, and he can utilize the creative mode, which is meant for creating the story. And he also utilizes the replay mode for previewing the story directly in VR. Then there is the observer, who can view the created content in the replay mode. And the last one is the camera operator, who utilizes the replay mode to select a recording, and the video mode to create a camera trajectory to export a 2D video recording. The camera operator and the narrator can be the same person in real life, but we distinguish those 2 user roles because it's different view from the user's perspective. So based on those roles, we came up with an application where the narrator can use the creative mode to create the story. The observer can use the replay mode, and the camera operator can create the camera trajectory in the video mode. We will look into those modes later. So there's an option to switch between those three modes directly in VR anytime. You can use the hand menu for that, and the replay mode is the default mode when you open the application, and you can select any already recorded scene here, and this option to switch between those modes is available in any mode. Okay, the next interaction that is available in the creative mode is loading objects. The user can click on any object and it gets imported. You can even import multiple objects. The narrator can also set up the scene by manipulating with the objects, rotating them, scaling them, and moving them around. When he's happy with the position of the objects, he can start recording by clicking the red button in the hand menu. And the user or the observer can then enter the replay mode, or even the story creator can enter the replay mode, and he can view the created recording there by selecting the recording from the recording list, and there you can see the avatar who is the representation of the narrator, and he's pointing to specific parts of the explained phenomena. You can also view the scene from any viewpoint in the replay mode. The observer can pause the story to interact with the scene and to explore the data. As the camera operator, you can enter the video mode and you can create the trajectory. You can see the preview of the keyframes above the keyframes itself. You can also see the preview of the video on the large screen or on the small screen in the hand menu, and you can see as you scroll through the frames that you can then click export the video that you previewed. In the evaluation, we formed two hypotheses. The first one is that the VRdeo tool will be easy to use and more engaging for the users than standard 2D videos. And the second hypothesis is that the VR environment will have a positive effect on the learning process. The first part of the evaluation, we conducted a case study with experts from biochemistry and medicine, also other fields such as geography, anthropology, psychology, and archaeology. And they were all overall very happy with the tool. We asked them to create short narration as in the role of the narrator, and they prepared the narration before the case study, and then we after the case study we discussed how they liked the tool and disliked the tool. The experts from medicine, biochemistry mentioned that they really liked the 3D models enhanced the understanding of the explain problem. They also mentioned that the playfulness of the whole process is another important factor that helps to keep focus. And also they mentioned that our solution would be very beneficial in the courses that they are currently teaching, and that it has potential to improve the quality and acceptance by the students. We also conducted a user study with 17 participants, and our goal was to compare the VR recording with the 2D video, and we had several questionnaires and a knowledge test. And the outcomes were very positive. We received positive feedback that the participants would like to use the tool as students, and all of them think that using virtual reality for education has benefits. So we measured that engagement when using virtual reality was higher, so that confirms the first hypothesis. And also that the observers in virtual reality had better score in the knowledge test than the users that were presented with the video. However, the results can be biased because of the group size. You can refer to the more details in the paper, but to point out one observation, we found out that it's beneficial to, it was good for us to use the stair color to visualize the state, the current state of the application, if the user is recording or the recording is paused or playing. And we visualized it by using the stair color because in desktop applications you can use simply a small circle somewhere on the screen, and that signifies that the recording state. But in VR we realized that we can use the stair color to reduce the movement of the head, in case the state would be signified only in the hand menu or somewhere in the scene. So in future, we would like to focus on the way how to edit the recording of the virtual environment, how to, for example, create a cut without losing the observer's attention. Also, we would like to come up with some advanced selection and highlighting methods, and automatic generation of the camera trajectory, which could really speed up the video creation process. So that's all for me. Thank you for your attention.
Yeah, thanks a lot, Wojciech. This was a really excellent talk, and the discussion just started. So please, everybody, join us for our discussion in the Discord channel. It's live right now. So we have already a good discussion going on with our other speakers from before. So sorry to say we are already over time, so we have to stop here. But please come to our second session, which will be also amazing and which will be headed by Jan Uska. So have a quick break now, and see you very soon in a few minutes' time. So we are starting at 11 with the next session. So take a quick break and see you there. Thanks a lot for joining today. Bye-bye.
See you, bye.